Hi, I'm Damon Smith, field crops pathologist for the University of Wisconsin-Madison. Today we're going to talk a little about introduction to plant diseases of field crops. So first I want to just talk a little about what a plant disease actually is. And a plant disease is any malfunctioning of the growth or reproduction of a plant caused by the continuous irritation by a primary causal agent that results in the development of symptoms. Now this is quite a mouthful, but I want you to understand that there's a couple key points here in this definition so that we understand how plant diseases actually cause malfunctioning in plants. The first is this key term, malfunctioning, and we need to be familiar with this because we need to know what plants actually look like in their normal growth stage. Sometimes plants will have what look like abnormal growth patterns but are actually part of that plant or supposed to be there. One sort of example of this would be in ornamentals, where we actually have variegation that is supposed to be on some of those plants. That is a normal thing that's supposed to be there, and so you need to be really familiar with the host so you can determine whether it's actually sick or not. The second key point here is this continuous irritation idea. And this is very important because this is what separates injury from disease. So injury is an abiotic or a non-biological cause of disease, and it often is acute. An example of this would be maybe off-target herbicide application that causes some injury to a plant, and there's really no progression of symptoms, okay, when we're dealing with maybe a contact herbicide in that case. This contrasts somewhat with diseases where we actually have a chronic sort of progression, okay? And so we have diseases which will infect, we'll get some lesions, those lesions will continue to spread and expand, and so there's this continuing process that happens with diseases. So you need to be out there looking at the crop and what's going on. Another key component of the definition is this idea of primary causal agents, and we can have the biotic and abiotic causal agents. The biotic or living organisms, these are the pathogens that we're going to spend most of the time in this session talking about. These are the organisms that actually have the ability to infect plants and cause disease. These are different from abiotic disorders, which are induced by non-living causes such as environmental conditions or things like herbicides and that sort of thing. Also, we need to be familiar with this idea of signs and symptoms when we're looking at plant diseases or trying to diagnose plant diseases. So identifying if a cause of plant disease is biotic versus abiotic is just half the battle. We actually need to identify the cause of the disease or disorder in a couple of steps here. So we want to actually, if we determine that, well, we're actually looking at a biotic issue here, or something that's actually being caused by a pathogen, then we would look for some signs of that particular disease here. So we're looking at signs of the pathogen, which are its parts or products that can actually be seen on a host plant. So things like pycnidia or fruiting structures would fit into this category. Symptoms are the internal and external reactions or alterations of the plant as a result of the disease. So this is the plant's response to the infection by that pathogen there. And these 2 points, while subtle, can be very important when you're trying to diagnose what's going on with a plant that's sick. Now, within the biotic group, we have 4 major pathogen groups which we talk about in field crops. And the first one, and probably the most important and what you'll spend most of your time dealing with, are fungi. And these are filamentous organisms that cause the majority of the plant diseases. These are commonly called molds, and they're usually microscopic, but occasionally their signs can actually be seen with the naked eye. They are much different from bacteria, which are single-celled organisms. They're microscopic as well, and you'll never really be able to see these with your naked eye, although sometimes you can see bacterial ooze in water with the naked eye, but those would be tons of cells that would cause that kind of disorder. And we're going to talk a little more about bacteria here in a few slides. The third category are viruses, and viruses are submicroscopic particles, and these are considered an obligate parasite, meaning that they have to be associated with a plant host or in a vector, either a plant vector or a parasitic plant vector or their insect vector to survive. Okay, so they can't be found outside of their host or that vector. And then the fourth are nematodes, and these are microscopic, non-segmented worms, which are also obligate parasites, so they have to have the host present to feed in order to survive. If they don't have a host plant there, they cannot continue to feed, and they will actually starve to death. So let's take a couple of minutes here to talk a little about fungi. This is a diagrammatic representation of what fungi look like here. They grow in this filamentous habit where they actually have this thread-like body which grows out, and the cells divide at the tips and move outward and grow across or inside a plant host. You can actually see the powdery mildew fungus here in this slide, or this image right here, growing out on the surface of the leaf. We can actually see the leaf veins here, and then the thread-like appendages of the fungus actually growing across the surface of that leaf there. So most of the time fungi are actually microscopic, but you can imagine when you have masses of this hyphae, which is this thread-like appendage, or thread-like body of the fungus, you can imagine that when it's in a mass of hyphae, or what we call mycelium, when it's in that mass, you can see that with the naked eye. Now, fungi are mostly spread by spores, which can be moved around by wind or even water or rain dispersed. And a couple of examples in field crops are going to be Sclerotinia stem rot of soybean, some of the ear rots of corn, and also Fusarium head scab in wheat. So bacteria, as I said, are a bit different from fungi. These are quite small, single-celled, spread mostly by water. However, contaminated seed and also insects can move sap that has bacterial cells in it. And bacteria have to enter the plant either by a wound or a natural opening. They don't have the ability to actually directly infect a plant like a fungus can. So a fungus can actually, the spore can land on the plant, we get germination, and the mycelium can actually directly penetrate the plant host. Bacteria can't do that, so we have to have wounding, such as, say, a hail event. And this is very important in corn when we're dealing with Gauss's wilt, because oftentimes we see fields that are damaged by hail having increased levels of Gauss's wilt because of that wounding event. Here's what a bacterium would look like if we had a microscope to see a bacterial cell. And bacteria spread through this process called binary fission, where we actually have division of the cells. So one cell makes two, two makes four, and so there's actually an exponential increase in the cellular load in a very short period of time. And this is why bacterial diseases can tend to spread very fast, especially when there's wet conditions around, which can promote spread of those bacterial cells. The third group, as I mentioned, are viruses. These are very simple organisms, and in fact sometimes we don't even consider them living, and there's some debate about that, but basically a virus is a nucleic acid, so it can either be RNA or DNA, and is simply surrounded by a protein coat. Okay, and they have to be mostly viewed using a fancy microscope called an electron microscope, and they do have some different shapes and sizes. Now, what's interesting about viruses is they have to be either associated with a vector or their plant host. And once the cell is inoculated with a virus, the virus essentially unwraps this protein coat, releases the nucleic acid, and that nucleic acid actually inserts itself into the DNA of the plant and essentially reprograms the cell to make more virus particles. So it's a pretty interesting process in how the infections take place and how the virus will actually manipulate the plant. Here's some examples of virus diseases. So we've got soybean vein necrosis virus on soybean here. This is a new and somewhat emerging disease in the upper Midwest on soybean. And then we have barley yellow dwarf virus here on wheat, a couple of examples that we typically see in field crops. Now, viruses will make plants very, very sick, but rarely do they actually kill a plant outright because they are an obligate parasite and they have to have that host around. It doesn't make a lot of sense for the virus to actually kill the plant, but they will make the plant awfully sick. And so a lot of times we'll see other organisms come in and actually kill the plants after the virus has compromised the health of that plant. Now the fourth group are the nematodes, and nematodes can come in a couple different types here. And actually we have 4 listed on this slide here. So we have the endomigratory parasites here. These guys, they will actually invade the root system and move inside the root. So endo means inside, and migratory means that they can continue to move throughout their entire life cycle. We'll contrast that a bit with the ectosedentary nematodes. Things like soybean cyst nematode fall into this group. And the ectosedentary, once the female forms a feeding cell on the root system, with her head penetrating the root tip, her body will remain on the outside of that root system for the remainder of her life cycle, and she stays in that one spot. So the sedentary means that they will not move any further after they establish that feeding site. Once the female is fertilized, the eggs will continue to form inside her body, and her body essentially becomes an egg sac. Once the eggs are fully formed, she will die, and then that egg sac can break open, and the eggs will then spill out into the soil for those juveniles to germinate and the whole process to start over again. The third group are the endosedentary nematodes, things like root knot nematode, which we deal with just a little bit in field crops. These are a little different than cyst in that the female, once she establishes her feeding site here, she is completely inside the root system here. Her eggs will form, and her body will actually expand, and as her body expands, that pushes out on the root, causing this gall that we typically see associated with root knot nematode. The fourth group are the ectomigratory nematodes, so things like sting nematode down south can be destructive on certain crops. However, here in the upper Midwest, we don't deal with the ectomigratory too awful much, but they can be present in some cases. These nematodes basically remain on the outside of the root system for the entirety of their life cycle and can continue to move throughout their life cycle. So they can cause quite a bit of damage, feeding on a root cell and then moving around, feeding on another one, and so on and so forth. Here's some examples of symptoms that we might see from nematode damage in a field situation here. So we've got some corn plants damaged by nematode feeding. We often see this sort of sparse emergence sometimes, or death in certain areas of the field. Here we have soybean cyst nematode causing stunting and some yellowing on soybean plants in this particular soybean field. And nematodes can be in an aggregated or a localized distribution in fields, and so you have to sort of do some homework when you're trying to diagnose nematode diseases on plants, because these can look like some other abiotic disorders such as water ponding and those sorts of things in fields. Now let's transition a bit here since we've talked about the major categories of pathogens, and we need to have a little discussion about the plant disease triangle. This is an important concept in order to understand not only how pathogens and hosts interact and disease develops, but also this can be useful when you're trying to develop a management plan for a specific disease issue. And so this is a pretty simple concept here, where we have essentially a pathogen, a host here which is susceptible to that pathogen, and both of those come together in an environment that is favorable for the pathogen to grow or compromises the host. So we have to have all three of these components coming together at the same time in order for the disease to develop. Now, If one of these components is missing or we reduce that component in some way, we could essentially reduce the amount of disease in that particular system. So for instance, if we do something here to manipulate the host component, such as deploy genetic resistance, say to soybean cyst nematode, we can shrink the amount of disease in that particular area by changing or manipulating that host. We could also manipulate the pathogen. We could deploy, say, a fungicide application or something of that nature to actually reduce the amount of pathogen affecting a host, and we could also reduce the amount of disease in that area as well. Or we could look at the environment here and do some things like manipulate the microclimate, such as reducing seeding rates in soybean for white mold. That can help reduce the levels of disease we see there. Or we could do something integrated. If we look at this triangle here where we actually manipulate the host, pathogen, and maybe the environment all at the same time, we can reduce the amount of disease even further. So you can sort of see how these components play a really integral role in how diseases develop here and how we can sort of manipulate these areas a little more, and we'll discuss how we might do that. So if we look at that host component again, genetic resistance is probably the most powerful disease management tool out there. And so when you're trying to develop your disease management plan, I would encourage you to look at genetic resistance and really study variety and hybrid trials each year in your area to choose things which have the best genetic resistance for the diseases you're trying to combat. Also use varieties that are well adapted to your area. Not only do we need to have some genetic resistance, but we also have to have varieties which are well adapted to the area so they aren't predisposed to disease. Plants can become stressed, just like you and I, and when a plant is under some environmental stress, that will predispose that plant to disease. And so you want to choose things which have the correct growing degree day units for your area, the right maturity groups, and those sorts of things. On the environmental side here, we talked a little about some things that you can do there, but here are a couple other things to keep in mind when developing your management plan. The environment can be very hard to change, but there are a few things that you can do. One of the biggest in the field crop areas is looking at soil fertility. Testing soil, making sure that nitrogen is in range, not too high, not too low. We also want to look at all the micronutrients and make sure that we have micronutrients in balance. So certain diseases can be worse again when plants are under stress or lacking some sort of micronutrient or essential nutrient such as nitrogen. So make sure those things are in balance before you start to develop your management plan even further. Also, plant population can be important. We can manipulate the microenvironment for certain pathogens. Again, one example would be Sclerotinia stem rot or white mold of soybean. If we reduce the plant population, that can reduce the favorable environment for that pathogen. Also, plant spacing can be important, and irrigation management. Most fungi like lots of wetness. Some bacteria spread around with heavy wetness or a lot of overhead irrigation, and so limiting irrigation only to only when plants need it is very important when you're trying to develop your disease management plan. Finally, on the pathogen side, there's a few things we can do here. We can adopt exclusion, where we actually prevent the introduction of a certain pathogen to an area. Probably one of the biggest things we can do here is choosing seed that is certified to be pathogen-free. A lot of seed for soybeans and corn and wheat will be certified, and that can be very helpful to keep a pathogen from even entering an area where it hasn't been present. Once a pathogen is in an area, we could try to eradicate it by removing that pathogen, although this can be very difficult once a pathogen has entered an area. So you can imagine, say, soybean cyst nematode entering an area. Once that particular pathogen is there, it's basically impossible to eradicate it. However, some foliar diseases could potentially be eradicated by actually removing some of the plants that are infected if you catch it early on. Probably most commonly used on the pathogen side is going to be sanitation and also fungicides here. So sanitation, where you're actually removing or burying pathogen-infested material or plant residue. An example here would be corn residue on the soil surface. Some light tillage to bury that material can help reduce the amount of foliar diseases when the subsequent corn crop is present. And then, of course, fungicide use is widely used now in field crop systems. Now, I want to take a little bit of time here and just talk a little about fungicides. We have a couple differences in terms of the types of fungicides we use in field crops. So we have what are classified the contact fungicides and then also the penetrant fungicides. The contact fungicides, some of the older products, things like Bravo or Dithane, are applied to the surface of the plant. They do not move on the surface or into the plant, and they can be readily washed off the plant. And they also have to be reapplied if a new part of the plant grows, because it will not be protected. Probably more commonly now in field crops, we have quite a few penetrant type fungicides. So things like Headline or Tilt would fall into this category. And the penetrants can come in a couple different varieties here. We can have local penetrants, which move into the waxy cuticle. Translaminar penetrants will actually move from one side of the leaf to the other side of the leaf. Cropital penetrants will find their way into the xylem and actually move upwards in the plant in the water potential gradient, and then finally the systemic penetrants, which will move upwards and downwards. Now, mostly in field crops, we have most of the modern fungicides we have are actually cropital penetrants or translaminar penetrants. We don't have any systemic penetrants in field crops right now that move upwards and downwards. So we're left with products which primarily move either across the leaf surface or up in the water transpiration stream. Now, here's a basic diagram of a plant disease cycle here. Okay, so we're using wheat rust here just as a brief example of how this might work in a particular situation and how we might use fungicides thinking about the plant disease cycle here. So what happens for many fungi is we have a spore which lands on that plant surface, it germinates, the hypha moves into the plant part here, you can see that, and then it forms a feeding relationship and begins to parasitize the plant. Now, a period of time goes by called the incubation period from the time that the infection takes place to when we actually see symptoms appear here. And this is a critical time point here in trying to make some decisions about actually applying fungicides. So our contact fungicides are primarily preventative. So they have to be applied prior to this infection process here. And so you have to have some ability to say, to predict when a disease might occur so that you can make that application of that contact type product. So you have to watch weather, keep good notes. The penetrant fungicides could be applied preventatively. However, if we think we've had infection or we see the beginning signs of symptoms here, we could apply some of these penetrants and maybe slow the disease down a bit. However, once we get to the stage here where we actually have sporulation within these lesions, this is really beginning to get too late for a fungicide application. So our window of opportunity for fungicide application is really pretty narrow, and we have to do quite a bit of scouting and keeping track of where we are in that disease progress. It's best to catch the epidemics early on to get the best control when we're trying to use fungicides in our plant disease management program. One final note on fungicides that I want to make you aware of is this idea of fungicide resistance. And so we need to be aware of this because we have a lot of single-site modes of action now in our fungicide market. And this is a good thing in terms of environmental stewardship, but it does place a tremendous amount of selection pressure when we use these fungicides widely across an area and repeatedly. And what I'm talking about here is I have a couple scenarios here in this slide where we have these little blue hyphae individuals here which are sensitive to a particular fungicide, let's say one of the demethylation inhibitors, which is widely used in field crops. And then we have one of these red individuals, which is a resistant individual in a population. Now what can happen is if we're not rotating fungicide modes of action, or we're spraying repeatedly, or we're using off-labeled rates, so these non-recommended fungicide use patterns, we can actually select for individuals in the population which are resistant to our particular fungicide. So after several generations, we can actually build up the population with resistant individuals here, and what will actually happen is we'll start to see our fungicide not being very effective in that particular field. We do not want this situation to happen. We would like to preserve this situation here on the left-hand side of your screen, where yes, we do have some resistant individuals which could pop up, but we keep the selection pressure towards the sensitive individuals. Okay, and so we're rotating different modes of action in our fungicide program. We're using the proper spray rates, proper spray volumes, and those sorts of things to preserve this situation. So here's some notes on managing fungicide resistance. Quite a few notes here on things that you can do. Of course, starting with resistant hybrids here is very important, looking at soil fertility. So these are some things that I talked about when we talked about the disease triangle, and how to integrate your approach here, and how to maximize the use of fungicides, and also to prevent fungicide resistance. And so you can begin to see how the integration of all these different management strategies can really maximize not only your management plan, but also your resistance management plan as well. Okay, so avoid sites with history of disease pressure, keep good field histories, make sure you're tank mixing high-risk fungicides with fungicides of lower risk. Don't use reduced rates. Apply fungicides preventatively. Do a lot of scouting to try to catch those epidemics early, and avoid any curative fungicide applications, especially with the high-risk fungicides out there. Also, make sure you're reading the label. The label is the law, and so it's very important that you're familiar with all the different regulations that go along with that particular fungicide that you're using. With that, I'd like to direct you towards my contact information. If you have any questions about any of the things that I've talked about in this presentation, feel free to email me at my email address. We also have a website here which might be useful for you. And also, of course, you can always get in contact with your local extension agent.